So this is Molly Gasparini. She has always been focused on human genetics-oriented research, beginning her research training as a University of Washington undergrad in Mary Claire King's lab. After a 2-year stint working in Louis Krunkel's muscular dystrophy lab at Harvard Medical School, she returned to UW to pursue a PhD with Jay Shinjuri in the Department of Genome Sciences. For her PhD work, she has been awarded an ASHG Charles J. Epstein Trainee Award and a National Science Foundation Graduate Research Fellowship. Welcome, Molly.
Great, thank you so much, Giovanna, for the introduction, and thanks to the 10X team for having me and for you all for having lunch with us. I'm going to speak to you today about a new genome-wide association framework that we've developed for cellular genetic screens that we've applied to tie enhancers to their target genes. And so our ASHG in this project is motivated by a huge problem in human genetics, and that is non-coding variants. So finding enhancers and understanding non-coding variants are, of course, really important in both rare and common disease. In the medical genetics clinic, it's estimated that median 10% of the time, though a patient is diagnosed with one of the best understood Mendelian monogenic disorders, that person's causal mutation isn't discovered by exome or clinical sequencing, one reason potentially being missed non-coding mutations. Additionally, it's estimated that 93% of common trait or disease-associated variants fall in non-coding sequence, but it's largely unknown exactly how these variants contribute to disease or affect disease genes. So of course, why are non-coding variants so tricky to understand? Well, even though 98% of the genome is non-coding, it's still largely unknown exactly where regulatory elements are and which genes they control. So even if you do find non-coding variants, you don't know if they disrupt a regulatory element or which gene that regulatory element controls. And this is tricky biology to figure out, of course, because though maybe we're used to looking at variants in linear space like this on the UCSC genome browser, in reality, of course, what is now understood is that in the nucleus, DNA is curving in 3D space, and these variants are sitting in a highly complicated genomic context. And so deconvolving this regulatory mess for every gene is critical to understanding disease, but it's, of course, really difficult just to figure this out just by observation alone. OK, so what data do we have so far? Well, one huge source of insight is human genetics. So by leveraging naturally occurring non-coding variants across human populations, the presence of a variant can be correlated with changes in any gene's expression in tissues from each person, and an association can thus be mapped between that non-coding locus and that gene. As you know, these associations are called expression quantitative trait loci, or eQTLs. And one use of them is to hypothesize that somewhere in this variance region is a regulatory element that controls the target gene. And this method is extremely powerful, as you all know, as it allows you to leverage one population of people in one data set over and over and over again to test all variants by all genes. However, of course, eQTL's insights, specifically into enhancer-gene pairings, are limited by linkage disequilibrium, as they can only study naturally occurring genetic variation, which falls in long haplotype blocks of many variants. And so this makes it difficult to figure out which variant is driving an effect, or if multiple are working together to do so, or sometimes there are no variants that fall into your candidate enhancer. So we wanted to take a step back and ask, what if we moved this framework into the pooled perturbation cellular screen paradigm? And so instead of human populations, we're going to leverage a population of cells. And instead of mapping with naturally occurring genetic variants, we're instead inducing large effect perturbations all around the genome using CRISPR. So after generating a guide RNA library and randomly distributing these guides in an unlinked way throughout the cell population, we can also leverage that same population of cells over and over again to test all guides against all transcripts and directly map enhancer gene pairs, which, since we were directly inspired by eQTLs, we've nicknamed these CRISPR-QTLs. So to note with CRISPR-Cas9, as you may know it, it's hard to make precise edits on all alleles. And so it's hard to create large non-coding effects with small indels that usually result from active Cas9. So just to start out and note, we are using a nuclease-inactive dead Cas9 attached to the Crab repressor to epigenetically silence and target candidate enhancers in the experiments I'll talk about today. Okay, and so our framework is working off of recent advance in CRISPR screens, where ourselves and others have applied CRISPR to interrogate candidate enhancers via pooled cellular genetic screens. So usually a library of guide RNAs are designed to non-coding sequence, and just one guide is delivered to each cell. And then the cells are physically separated based on expression of that one gene to phenotype each cell and thus each guide. However, these methods are only testing only testing each candidate enhancer against one gene at a time. And so in reality, doing this for all genes is going to be quite laborious. And so instead, ideally, we want to be able to test all enhancers against all 20,000 genes or all genes expressed in your cell type of interest. So one obvious method to do this would be to induce enhancer perturbations individually and perform bulk RNA-seq. But this is obviously going to be very slow, and we're at the 10x Genomics luncheon. So a faster way would be to adapt single-cell RNA-seq to this pooled CRISPR screen format. And fortunately, recently ourselves and others had developed techniques to simultaneously capture the guide and single-cell RNA sequencing data prior to the work Giovanna described. And so each guide can be simultaneously tested for control of all genes at once in each cell. And an approach like this was previously applied to the non-coding genome and was published by Gary Hahn's group last year. And there they tested 71 sites and 7 topologically associated domains with just a single guide per cell. However, single-cell transcriptomes are still costly. And so at this throughput, it's still prohibitive to test all enhancers against all genes. And they captured one whole transcriptome to phenotype just a single non-coding perturbation. So what we realized is that since each guide is only targeting a small amount of real estate, we can multiplex the number of guides per cell without ever really targeting the same site at once. And so the more we can multiplex guides, the more power is gained for each pooled screen experiment. And we get much more efficient use out of each single cell transcriptome by using it to phenotype multiple perturbations at once. So mainly if we target a large library of sites across the genome, when randomly drawing 15 or 30 of those to package per cell, these sites are going to be likely targeting very different parts of the genome, so we can analyze them independently. And we're enabled to do this by borrowing from the QTL analysis framework, and so we can correlate presence of a perturbation with presence of a transcript's phenotype. Okay, so we posted this pilot screen of this work on bioRxiv in the spring. And there we targeted about 1,000 candidate enhancers, which was almost 15 times more than the single guide per cell RNA-seq non-coding screens that were so far published. These sites are distributed around the genome, such that overall we tested about 17,000 potential cis enhancer gene associations. And there we delivered these guides at 15 per cell and collected 47,000 cells to test them. And we posted this on bioRxiv, but today I'll also tell you about a new scaled-up screen of 5,800 sites, which is about 80 times more than published screens. And there we performed about 78,000 gene enhancer association tests. And here we delivered 28 guides per cell and collected 207,000 cells to test them. And to note, just from start to finish, these experiments take about 6 weeks if everything goes well, with mainly just me working on them. Although for this big screen here, I did get a good amount of extra help just to help me collect this number of cells at one time point. Okay, so I'm going to tell you mostly about this unpublished scaled-up screen today. So we performed all these experiments in the erythroleukemia cell line K562. And there we picked candidate enhancers as defined by sites of open chromatin that were not within a KB of coding sequence and were largely enriched for other enhancer-associated marks, including transcription factors important to blood development. And then we designed 2 guides to each enhancer to account for guide RNA quality. And so then, as Giovanna described, we took the guides designed to the 5,800 sites, and we made lentivirus with them. Then we infected these at high multiplicity of infection, such that there were about 28 guides per cell. And then we collected the 207,000 cells via the 10x Genomics V2 single-cell RNA sequencing platform. And so at the time, guides couldn't be innately captured in single-cell data, but fortunately, tech had already existed to capture them in each cell. So we used an optimized crop-seq expression vector from Christoph Bach's lab with a capture protocol that we had optimized to sequence and assign guides per cell with high confidence. And so just to note from this data set, this meant that each site was targeted in about 900 cells. And so to get the same power with just one guide per cell framework, this would have required almost 6.5 million single-cell transcriptomes. Okay, so now we have these cells, their guide RNAs, and the transcriptomes. So to test each perturbed candidate enhancer for any potential target genes, we again in silico separate out the population of cells based on guide RNAs targeting one site at a time. And then between these two in silico cell populations tested for differential expression of all possible cis target genes using